Oxidative phosphorylation. Oxidative phosphorylation follows all the pathways from the glycolysis through pyruvate oxidation and the citric acid cycle. It is a way that we will utilize the reducing elements NADH and FADH2 to create even more energy through a membrane potential. So oxidative phosphorylation is also known as the electron transport chain. As we will see, it transports a lot of electrons. So it occurs in the mitochondria. Its proton gradient is established over the mitochondrial membrane, which then induces ATPase to make ATP from ADP. It's a combined redox reactions, and well, as I just mentioned, it occurs in the mitochondrial, well, the inner mitochondrial membranes, I should say. And this is where O2 is reduced to water. So this is the first time that we actually utilize the oxygen that otherwise have been not present in anywhere in the general metabolism. So the membrane potential, I think I've mentioned it before, but just to reinstate here, membrane potential is the driver of all living chemistry. The membrane potential enables us to drive a proton pump that create a rotation that will create turn ADP into ATP. And as you can see on this slide, we basically have a proton pump inwards to the mitochondrial inner membrane or inside, and then we will have a proton pump that pumps the proton out again of the mitochondria, and this creates a proton gradient. Just to recap, the photosystem in the chloroplast acts exactly the same way. So here, light is inducing the photosystem 1 and 2 to pump protons into the thylakoids, and the ATPase then produces the ATP. So there you see it's the same principles. It's many of the enzymes are the same in the two pathways. This proton gradient can actually be established by chemistry. So one can show that you can induce ATPase functionality by changing the pH on either side of this membrane. Likewise, as we will see, the citric acid cycle is fully connected with the electron transport chain via complex 2, which is basically, complex 2 is basically the same as the succinate dehydrogenase. It's two different domains of one enzyme that is physically bound together. So the succinate formation of fumarate creates this FADH2, which in the complex 2 will be utilized in the quinolol complexes. So this is the physical way that the citric acid cycle is connected to the electron transport chain. Now, where in the cell is this going on? No, we know about the mitochondria, and the mitochondria outer membrane is a quite open membrane that allows a lot of molecules to come in and out, whereas the inner membrane is a bilayer lipid membrane where you can have complexes sitting like we see here in the membrane, and you have an outer and an inner surface. The space in between is called the intermembrane space, not to be confused with the matrix. So the matrix is actually what's inside here in the mitochondria, and then the cytosol is around here. So what we see here is a depiction of the inner membrane with the electron transport chain complexes here, 1 to 4, and the ATPase over here. So this was a eukaryotic cell. In the prokaryotes, as you know, there is no mitochondria. So where is it situated here? Well, first of all, we have the subunits of the complex 1 dehydrogenase, which is situated up here. We have a couple of the complexes actually linked together in this area, as you see up here on your right-hand side. And then you have the ATPase down here on, again, sticking out from the cytosol of the bacteria into the intermembrane space here. So it's exactly like in the mitochondria. It's just that instead of having a matrix, you have a cytosol in here, and then you have the outside of the cells here. So that's where the two parts are. So it's basically situated in the same place. Now, reductions and redox pairs. What do we have for us to come here? We have a lot. We're gonna concentrate a lot of the NAD plus redox pair with the NADH redox pair. So it's a two electron transfer, and as you can see here, all the top here are reactions that donate electrons. So if we go from the reductant to the oxidant, then we donate electrons. Where down here, it's an acceptance of electrons. So we will be, as I said, focused on this pair and of the FADH2 pair. So basically acceptors in the beginning and donators later on. We will also touch upon the cytochrome C, or the cytochromes in general, that have a heme binding site. And the heme binding site is chelated with ions, and this is one of the ways that we will oxidize NADH into NAD. Ubiquinone will also be mentioned as one of the oxidized forms that we will work with. Likewise, we have some thermodynamic data that will again here indicate what is it that's going on. And as you can see here on this slide, now it's depicted with NAD plus to NADPH. So that reaction, the other reaction, have a favorable electron flow in this direction. So if we have NADH, we really want to get back to NAD plus. So that's a more favorable. So if you can donate those electrons to another system that could be heme, pyruvate, or some other stuff, well, then that is a preferable reaction. We can see here that ATP, the range of hydrolysis in this electron negative area, and exactly where we have this change with the NADH to NAD. So that fits very nicely with each other. So with the knowledge of the electron transports, or the electron, I should say, flows that we want to study, this table just shows you what was shown just before. And as you can see in the very, very bottom, there is the reduction of oxygen to water. And this is where we want. This is what we want to happen. We really want to utilize the electrons that are captured up here in the ADH to be turned into water. So this is the overview table, and with that, I think it's time that we go on to hear about the complexes that are part of the electron transport chain. So that will be the next video.